Hello and welcome to this week's What Were They Thinking? Among this week's news items, we have exciting news for epidemiologists. The first death from H5N5, better known as bird flu. A daily supplement may help boost brain function in older adults, with a great deal of skepticism. We have a diabetes drug that 60 years later we've discovered, which may yet prove to be a very detrimental result. Nasal drops used to help with brain cancer. Our ancient ancestor Lucy was neighbours with an even older human ancestor, which kind of makes sense, she had to come from somewhere. And a newly discovered organism could represent a new branch in the tree of life, creating all sorts of problems for biologists and students who learnt otherwise in school. The timestamps for these and other news items can be found in the description box below. Let's start with the good news, bad news. Good news if you're an epidemiologist. You have just found that H5N5 or bird flu is actually able to kill humans. This means all sorts of research funding and other opportunities are about to come up. The bad news is that for everybody else, we're about to have another pandemic. Yeah, this is not good news for most people. But then again, for those who it is good news for, they're going to be really excited until they get bird flu themselves and die, in which case they're probably going to be sad. Or maybe even more excited. It's hard to say with epidemiologists sometimes. This comes out of Washington State in the United States of America, wherein an individual contracted a rare strain of bird flu that had only been thought to infect animals. Well, technically we are animals, so this is not that surprising, but non-human animals. State officials have said that it's the second fatality linked to the United States and bird flu this year. So this is not a fun time for anybody. And the good news, and we say good news with a great deal of hesitancy, is that they were an older person with underlying health conditions. So, in short, they were already kind of damned. They had enough health conditions and old age already that meant that they were predisposed to dying at a stiff breeze, let alone if they encountered an actual proper disease. This death was confirmed by tests conducted by the University of Washington, and yes, it was definitely bird flu, or avian influenza as it's also called. So, yeah, this is not a great result. Slightly better side to this is that the risk, let's call it, to the public remains low. But low is not none, and therein lies the problem. The let's say, theorized vector for infection was not wild birds, which is a concern as well. Instead, it appears to have come from the flock of chickens, mixed breeds of various domestic birds that they kept in their own backyard. And it would appear that in feeding and caring for those chickens and other domestic birds, that they were infected with bird flu that way. And that is a concern, as that means there's now a much more, let's call it, a viable vector for infecting more humans, and if that happens, we are in trouble. And next we have good news, that is almost purely good news. First of a kind that boosts vision in humans, at least according to the clinical trial. The condition that it treats is called macular degeneration, and that's when the central field of sight becomes blurred and eventually you can no longer see at all. Rather than looking through opaque glass, it's just you can't see, full stop. Current treatments basically slow the progression, and if you can't slow it, you try and make your vision better. But the blurring will get worse over time. The treatment itself, as it is now, uses stem cell transplants, which stem cells are basically the fix to everything. It's the duct tape equivalent of medical care. You slap a bit of duct tape over it, and suddenly, hey, look, it's fixed. You slap some stem cells in there, and hey, look, the thing has been returned back to normal.
How long was I in there?
About 5 minutes.
Why are we not funding this?
The slightly facetious description, though, is accurate, as the stem cells are able to safely reverse the cumulative damage to the macula, and therefore return vision. The first of kind, let's call it, has been tested in humans in a phase 1 and 2A clinical trial. The main focus of this is effectively, can we do this safely, because it's all well and good if you do something, but it has a 50% fatality rate or worse, and it's also kind of important to know that it actually works. If everybody dies, well, that's bad. If half people die, but the other half get their eyesight back, that's better, but still very bad. If 99.99999% of people happen to get their vision back and survive, that's probably okay. Of course, you really want to know about what's going on with that fraction of a percentage. The results are good except for one fairly major caveat. There were 18 potential patients involved in this study. So that sounds like a really small study, but hey, it's only phase 2, except that they narrowed it down to only 6 of those 18. Yeah, it's a problem in those terms because it's a really tiny study. The participants involved had anywhere between 2200 and 2800 vision, which is not great. Admittedly, the others were marginally better, marginally in that they can see a bit better with 2070 and 2200 vision. Good news in terms of this treatment going forwards, and this is very much a good news, bad news situation, is that it does rely on stem cells, but those stem cells come from an eye bank that can basically produce the new cells as needed. This does mean that we could theoretically roll out this treatment on a much larger scale, and given that it's shown to be safe in 6 individuals, they're probably going to move on to further stage 2 trials, but if this does continue in the current trend, that is a very promising treatment to at least the dry form of macular degeneration. Going on now to other treatments, this one could theoretically be more useful to a wider portion of the population. It's a twin study, which is probably one of the best kinds you can possibly get, in which you could take a daily protein and prebiotic supplement. Now, the catch here is that it's looking at older people, and only looking at improving memory test scores.
So I tied an onion to my belt, which was the style at the time. Now, to take the ferry cost a nickel, and in those days, nickels had...
So this is arguably good. So then they took this same idea and went, okay, what if we run a double-blind trial? They used plant fiber prebiotics that are available over the counter in many countries. Now, prebiotic non-digestible consumables are basically dietary fiber. It makes your poop solid and means that when you poop, you don't give yourself hemorrhoids, or possibly detach a part of your colon and have it prolapse. What the researchers have done is take a dietary fiber in the fructan class. This is called inulin, not to be confused with insulin. It's used as a low-calorie sweetener in foods. And by using this, they were able to look at whether or not the people consuming it had an improvement in brain function. Again, caveats though. Very small test size, 36 pairs of twins, which, even though it's smaller, is actually much better because it is a twin study. However, you must take a twin study with individuals like this that are all at least 60 years old with a great deal of skepticism, because that's 60 years of divergent changes in twins. Unless you have 2 twins that have basically lived side by side for their entire life, this kind of twin study at this age is helpful, very helpful, but it does have notable limitations. Each of the twins was split. One received the daily prebiotic mixed into protein powder, while the other received just a placebo powder. And, well, they basically then took their memory tests and said, okay, what's going on here? And, yeah, it works. The basic idea here is that, hey, it works in mice, maybe it works in humans. And yes, it does work in humans apparently, or at least older humans. And that is perhaps one of the bigger concerns here. It appears to work in humans by providing them with a, let's say, usable source of energy in the form of this inulin that's not necessarily, so widely usable by the body like, say, glucose is. The benefit to this is that, unlike, say, pure glucose, which would contribute all sorts of issues to diabetes, this doesn't have that effect. And of course, it does have various effects on, say, producing solid poop, which particularly for the elderly is a concern. They are more prone to prolapsing various parts of their body. In other neurological news, we have claims that a gut bacteria is linked to Parkinson's disease, and therefore theoretically provides a treatment option that's relatively straightforward. This is a interesting claim. The argument links the development of Parkinson's disease with the production, or is it perhaps absorption, of riboflavin, biotin, and other B vitamins. These B vitamins are also somewhat produced by, well, partially bacteria in the GI tract that produces them as a metabolic byproduct. The theory is that by supplementing B vitamins in individuals, you could theoretically achieve the same therapeutic outcome as you would for somebody who has the bacteria required in their GI tract. The challenge, let's call it, with this research is that, well, you really have to sort of treat people really early on to be able to get it to work. The second is, that it's based on poop.
It's like as soon as people learn they have microbiomes, the first thing they want to do is start swapping them.
Well, young man, the truth is we still don't know a lot about the microbiome, but you shouldn't let it freak you out.
But where does it stop? Then are people just going to start wanting skinny people's microbiomes to be skinny, or an athlete's microbiome to feel athletic and young again?
Yes, it's possible, but we don't know enough.
Fecal samples from 94 patients with Parkinson's disease and, arguably, thankfully, 73 controls, although that does mean some poor postdoc had to go rummaging through feces, found that, yeah, this is basically the link they've got for now. The good news on top of what we've mentioned, which is there's actually a treatment pathway, is that it's a relatively benign treatment option, and that theoretically, through dietary changes, you might observe the same impact in selecting for microbes that will produce the same bacteria within your GI tract. The benefit to this is that you don't necessarily need to have the bacterial change. You could theoretically provide B vitamins, and those B vitamins would then help with the production of, say, short-chain fatty acids and polyamines. These would help in producing an environment more conducive to not getting Alzheimer's disease or Parkinson's disease in this case. The benefits to this is that by, let's say, exploring this in greater detail, the researchers have sort of been able to demonstrate, but nothing definitive, that there is a protective layer of mucus in the intestines, and this helps with the intestinal nervous system, and by helping to bolster this protective layer, some of the toxins we encountered through digestion could then have a negative effect on those nerves, and these nerves then carry issues all the way up through the rest of the body. This, of course, is very interesting, but not necessarily anything definitive, as, well, it's basically a somewhat observational study. Now, if they were to try this in a clinical trial setting, maybe. Basically, they would get a group of people and go, here, consume these prebiotics, and possibly in turn containing the microbes required, or in other cases just, here, take these pills with B vitamins, and they would be able to figure out what's going on. Maybe. Going on now, we have new and more fun drugs. Metformin has been around for a long time, which is good. It's kind of important. It's used by a lot of old people when it comes to managing diabetes. But after 60 years of being around, it has an effect on the brain that we didn't realize was there until now, which is a concern. One of the ways metformin works is that it lowers blood glucose by reducing glucose output from the liver. Basically, you can't really stop absorption from the GI tract, but you can stop, to some extent, how much of it gets released back into the body. And so they try to figure out, okay, this is going on, but what else is happening in the body? And, well, they found that the brain plays a pretty big role in that regulation, which makes a lot of sense. The tests they're conducting now have basically tried to track what goes on when you give mice metformin, and they found that by giving them this drug, it travels to part of the brain, an area called the ventromedial hypothalamus. There, it helps deal with type 2 diabetes by turning off a particular kind of, essentially, a protein. It's a little different to that, but let's just call it a protein to make life easy. This protein is called RAP1. When RAP1 is present and they give them metformin, the mice appear to be better off. When you take away RAP1 by genetically modifying mice, you basically have, no effect whatsoever. So clearly, there's something going on there. And that alone isn't all that helpful, but it's what they did next that proves to be more useful. What happens if you give metformin directly into the brain? They found that not only is it long-lasting and effective in the brain, but that neurons that are activated with metformin are, let's say, better off than they would be without this, in very broad terms. So now it has two mechanisms of action. Obviously, we had the liver, and now the brain. The next thing we just need is the pancreas, and we've basically hit the trifecta of things to worry about when it comes to metabolism, absorption, and function of most hormones related to digestion. In further news related to diabetes, we have new drugs. This new drug would be an alternative to, say, Wegovy and Ozempic. It is a glucagon-like peptide.
FDA-approved Lizzo makes you feel good about your weight, and it costs 90% less than Ozempic.
The new clinical trial involved about 1,600 people, so it's relatively small level, reasonably early in the stages so far, but it works just as well as injectables for people with type 2 diabetes. The drug is called orforglipron, which sounds like it should be some sort of Lord of the Rings character, but still. The experiment was conducted over a long time frame, which is helpful. It was nearly 1.5 years at 72 weeks. It involved 136 sites in 10 countries. The team was led by obesity specialists, and people taking this pill lost nearly 10% of their body weight on the highest dose of the drug. Now, this is not necessarily a bad thing, and it's basically comparable to people taking things like Wegovy, Ozempic, so on, who lose somewhere between 10 and 15%, but remembering they need to inject their medication, whereas this is an oral pill, you swallow it and you're done. No concerns about, say, having to deal with needles, a possible infection from not cleaning the injection site properly, or other contamination, so on and so forth. The good news is that it also follows the same effectively mechanisms as other glucagon-like peptide 1 drugs, and therefore has a similar safety profile. Knowing how it works already having drugs that have been approved, and so on, basically means that if this is proven to be as effective as it appears to be, and there's a big caveat to that, then arguably it's going to get approval much more easily and much more quickly. If it proves to be that way, though, that's the key here. What makes this drug viable, let's say, is that unlike the other injectables, Ozempic, Wegovy, etc., this is not a peptide, and that's an important distinction. If you were to consume a peptide in the way that this pill is being taken orally, you would have lots of problems. The stomach acids would break it down, the proteases would then further break it down, and basically it wouldn't work. By working the way this does, it essentially binds to the same receptor as these peptides would work with, but it isn't a peptide. In similar drug development news, we have what is probably, possibly, in many years' time, good news for those with brain cancer. And not so much if you're brain dead like RF cray cray and have to deal with brain worms, but brain cancer at least is promising. It's a possible treatment that uses the nose as a delivery vector. Understand, brain cancer is an absolute nightmare to deal with, and not just from a patient perspective, but from an oncology perspective. The reason for that is that it's very hard to get drugs into the brain. The blood-brain barrier is very, very resistant to most treatment. This means you need lots of high-dose treatments that have all sorts of side effects and can cause all sorts of other grief. Second is that a lot of the cancers that develop in the brain are a bitch to treat. They just don't die. Third and finally, well, if you do treat them, that part of the brain is gone. The brain does not regrow once you take something out, unlike, say, the liver. And you don't have a replacement like you would with the kidneys or, let's say, parts you can afford to lose like the GI tract. This brings us to the actual treatment they have, which is avoiding many of the initial issues, that is, delivery and dose. The nasal drops that travel along the nerves in the nose straight into the brain, which allows it to directly address brain cancer. Understand the olfactory bulb, that is the great big nerve in your nose that detects various smells, odors, etc., and allows you to know that somebody has farted, is also directly linked to the brain. It's a nerve that goes straight from your nose into the brain, and therefore if drugs are traveling along that, or for that matter other diseases, you get straight into the brain and avoid the blood-brain barrier. But this study is based on mice. So we basically think, okay, this is cool, mice are great and all that for research, but will this ever actually translate? And therein lies the biggest barrier to this treatment option. We're not saying it isn't possible, but it is theoretically a possible candidate, but a candidate with huge caveats. The treatment they're looking at, though, is specifically for a form of brain cancer called glioblastoma. and, well, other, let's say, treatment-resistant cancers. If this works as intended, it would be groundbreaking, if not redefining. One of the reasons why glioblastoma tumours are, well, hard to treat is that they're aggressive, hard to detect early, and essentially turn off all of the immune system response, meaning that most of the typical approaches just don't work with them. You can't shrink the tumour and have the immune system take care of it. They're aggressive, which means they grow quickly, which means surgery is very dangerous, and if you don't get them early, you're basically cutting out the majority of someone's brain. So yeah, very hard to deal with. But if, as this treatment would suggest, you can treat them directly, and that's through by virtue of, say, these nasal drops, you would be able to reduce the severity of the cancer and theoretically being the key word here, shrink the size of the tumour and allow for surgery that would have less impact on the brain. The treatment itself isn't exactly new. Rather, what it does is it stimulates part of the body. It's the interferon genes, and this allows for the body to effectively initiate a sort of inflammatory response, saying, hey, you've already looked here, but come back and take another look because there's something here that's not right. Usually it responds or is activated in response to viral infection, which cancer kind of sort of is. It's not, but it's a similar sort of idea. And normally, giving this sort of treatment requires multiple administrations, those administrations of the treatment being very invasive, and, well, you have to do that because the treatment breaks down very quickly in the body. This administration route mitigates just about all of those issues. You don't need to administer anywhere near as much, anywhere near as often, and you don't need to be invasive when doing so. The only concern we have with it as it's designed now, and this is a somewhat more of a very niche complaint, is that the researchers had the genetic material turned into a circle plasmid basically, and then wrapped this around a gold nanoparticle core. Now, depending on the volume of gold used, it's going to be very expensive, which could be a concern, although that's slightly less of a worry than, well, possibly building up heavy metals in the form of gold in somebody's brain. But if the treatment does work as intended and you don't need that much and for that long, this should actually be a fairly successful treatment pathway with low risk. And going now to sleep, and possibly that bacteria in your brain are making it harder or better for you to sleep. And, well, yeah. The Maslow's hierarchy of needs has sleep as one of the major requirements. The generally accepted view is that sleep is basically dictated by the wider world. It's a social thing, not a biological imperative as such, or at least when you sleep. The need for sleep is definitely biological. But a study produced in Frontiers in Neuroscience has claimed that actually bacterial cell wall components, primarily peptidoglycan, have been found in parts of the brain, mostly the brainstem, olfactory bulb, and hypothalamus, and that this would likely have some degree of effect on sleep, or at least our want for sleep and ability to sleep. Peptidoglycan is the thing in the cell wall of various bacteria. Some of them have a thick cell wall, some of them don't. If they have a thick cell wall, they're peptidoglycan positive or gram positive. If they don't, they're gram negative. And what it does is provide a very strong structural component around the outside of the bacterial cell. This ensures that they can survive in most environments without being broken down due to all sorts of different things, whether it's osmotic pressure, environmental exposure, and so on. But this is where the research is interesting, because we shouldn't be finding peptidoglycan in the brain. The brain should stop anything like that from getting through, because it would require bacteria to some extent, or at least bacteria that are being destroyed at some point. So how is it that we're seeing signs of something that should be in your GI tract in your brain? Well, to start with, we're not seeing it in human brains. They're looking at mice, and they're looking at a very small number of mice, which is a huge problem. Just 9. 9 male mice. Now, they were kept in the usual, what's the word for this, conditions, let's say? A 12-hour light-dark cycle, and they took measurements over 48 hours to map brain activity during the cycles of sleep and rest. This seems like it would be interesting and useful, but basically such a small study is like saying, hey, look, that cloud looks like a dog. Cool. Do all clouds look like dogs? Don't know, didn't look at them. See how this could be a problem? This is why we take such research with a lot of skepticism. It might be something, it's also very likely to not be something with such a small study size. Remembering, 9 mice isn't even statistically significant. It's enough that an honest student might be doing an experiment, and that's what they published this on. Moving on now to older animal news and human news, we have Lucy, one of the ancient human ancestors. Basically the missing link, if you will, somewhere between apes and humans.
If your elitist East Coast evolution is real, why has no one found the missing link between modern humans and ancient apes?
We did find it. It's called Homo erectus.
Then you have proven my case, sir. For no one has found the link between apes and this Homo erectus.
Yes, they have. It's called Homo habilis.
Aha! But no one has found...
It would appear that Lucy was actually living near even older human ancestors, although older human ancestors is going to be a bit of a challenge, mostly because there's not a lot of this ancestor actually available. We say not a lot, as in there's a handful of bones that don't match with what's going on, and therefore had to be from another species. The 3.3 million year old remains in Ethiopia's Afar Rift are an important discovery as they show human evolution over time, but also, they are partial remains. What these remains provide, other than showing that yes, there are older humans, and that, humans, hominids might be a better word for this, and that they were different again, is a difference in how they would move, the locomotion. The jawbones that we discovered previously weren't that much help. They certainly showed evolution and change and different species, but they weren't quite definitive enough. These feet from Afarensis suggest that the two coexisted together and that they had different lifestyles. Lucy would have been an expert tree climber, which is helpful for escaping from predators and possibly early hominids that were looking to kill her and eat her. The big toe and other foot bones suggest that the other, this being Demerendia, were adapted to not just climbing trees, which is what we'd expect. Rather, it was also able to hyperflex, meaning that it was able to walk comfortably on 2 legs as well. It's a hybrid between the arboreal adaptation, what you would expect of most apes, or at least monkey-type apes, and what you would expect of, say, humans or other apes that travel on the ground more than the trees. Carbon dating within both species shows that they had very different environmental conditions under which they would evolve and adapt and be able to grow. In this case, Lucy was able to be more diverse in the food she ate, whereas this new discovery would appear that it favoured eating foods that would grow in cooler, wetter climates, which would be helpful if you were in a forest or a rainforest of some description, but because of this and the fact that, the world was changing at the time, the ability to consume such food would have been limited and getting worse over time, allowing for environment, that is nature, to kill another species. Mother Nature's hobby is somewhat morbid. Now we have news on ants, and not just because ants are interesting and weird, but that a parasite might be taking control of them and forcing them into weird slavery. The way this parasitic organism works is that it causes them to kill their own queen, a regicide being another one of nature's weird hobbies, but less intentionally so. The way nature has crafted this particular situation is somewhat creative and, again, nasty. It involves a parasitic queen from either the Lasius orientalis or L. umbrotus species, who gets into a colony of other ants, either L. flavus or L. japonicus, and essentially causes the ants within that colony to kill their own queen, and then place the new queen from a completely different species on the throne. Yeah, this is nasty, and the whole reason they can get away with it is simply that, they're able to create the necessary pheromones and other signals that are used to communicate, and from that, trick the ants into thinking, hey, she's not the queen, this other one is, and therefore recognizes the original queen as the enemy. This allows accrue to be conducted, and regicide to follow. To prevent the original queen from being identified and recognized as being a legitimate monarch, let's call it, the parasitic queen will instead spray the original queen with formic acid. This prevents the queen's normal scent from being available to the ants to detect, and instead they think this smells nasty and foreign and get rid of it. Going now to other news on life, we have a new organism that could be a new branch in the tree of life. Could be being a very significant caveat here. It's a marine ciliate that scientists have been tending to since they found them in Croatian waters in 2011. The weird thing here is that although we've had them for over 14 years now, the organism itself wasn't really clearly understood because they had to wait until the ciliates died. The death of the marine ciliates is what led us to the discovery of this new bizarre organism, and, well, it looks like an alien of sorts. Almost bizarre in the way it's structured, and we say that in sort of some respect for how messed up it appears to be. It's a Frankensteinish creation, which we're impressed by. This is a single-celled eukaryote. This means it has membrane-bound nucleus full of DNA, just like our cells, and it has mitochondria, like our cells, which is helpful, but it doesn't fall within any of the current existing categories for eukaryotes. Beyond just being a weird new creature to add to the phylum, what we have is genetics that are rather more interesting in terms of evolutionary biology. The discovery, let's call it, of a gene called SEC-A. This is a gene responsible for getting proteins across a membrane. Now, this shouldn't be important for mitochondria because mitochondria don't really need to worry about that. They have portals and they have everything else provided by the cell they're in. But if mitochondria were originally a completely independent organism, then this would be necessary. And this is where SEC-A proves, at least more definitively, that it was in fact part of a, well, in this case, a previously independent organism. Further to that, the organism that we found this gene within is kind of funky and looks almost like a little solar system, which makes it even more impressive. Shifting to more environmental news, but not environmental news exactly. We have rare earth elements, that stuff used for fancy electronics, batteries, solar panels and so on, found inside plants for the first time. Now, this is not necessarily the most important discovery just yet, but theoretically, the fern called Blechnum orientale could be used because it collects and stores rare earth elements. Consider this, rare earth element mining at the moment is, well, let's be blunt about it, incredibly destructive, and not just to the local environment. It causes all sorts of damage in that respect, but importantly, it also kind of fucks with everybody who lives in that area. The miners get sick and die, the environment is poisoned, and, well, big businesses take over and cause all sorts of problems in that respect. So, not cool. This is where the fern might provide a more sustainable way of, we wouldn't say mining, but certainly gathering the mineral resources that society relies on ever more and more every day. There are 17 rare earth elements in total, and basically these are all metals that are found in every kind of technology you can imagine. And this is where possibly, but certainly not exactly ideally, the earth might be mined, let's say, using these ferns, and it would be far less destructive. Possibly anyway. It's a concept called phytomining. Essentially what we've described, which is take something that's known as a hyperaccumulator, or a plant that can collect this mineral metals in this case, in otherwise soils rich in the material you're after, and by pulling them out of the soil is left uncontaminated, or at least without these metals that we want, and we basically take the plant and process it in such a way that we then get the metals we want, or anything else you're after. It's similar in some respects to, say, how you might use algae to make oxygen, how you might, say, remediate soil using certain bacteria. There are a variety of ways in which it can be used. And so this is where the, well, obviously, usefulness of this fern is particularly important. In other sort of plant-related news, but certainly going much further afield, we have moss, and not just moss, but moss that has managed to survive 9 months in space. Now, moss is very resilient. It can survive a lot of shit, and I mean a lot. It is surprisingly resilient to just about everything to some extent. It can often be very readily dehydrated, and then after dehydration, it will come back alive once you rehydrate it. And, as it would appear, it can survive harsh radiation exposure. We know this because it's been stuck in the vacuum of space for those 9 months. The researchers took spore-containing capsules of a species, and this species was called Microbiome patens. This is also known as earth moss. By leaving these spore-containing capsules in space, they basically just waited to see what happens after 9 months. And they found that, yeah, it works. It works extremely well. It survives. Admittedly, it doesn't survive perfectly, but, many of the organisms that should have otherwise died, in this case the moss, have lived, which, given that this is space, it is cold, it is obviously dry, and there is an arse ton of radiation that should cause all sorts of problems, the moss has come back very well, all things considered. To use a very rough comparison, a human in space is liable to develop all sorts of complications just from being in space, and that's in a protected environment, let alone being exposed in a somewhat vulnerable way to the outside of the ISS. They probably wouldn't survive very long in that case. Their blood would boil and they would die. If they were in a protected situation, they would still experience radiation exposure, meaning cancer at best, and of course, 9 months of that would be death. They just would not manage. Interestingly enough, the fact that they returned those spores to Earth after 9 months and the spores worked was already helpful in understanding just how resilient the species is, but it's the other changes that were a little more curious. The researchers found that there was a 20% reduction in levels of chlorophyll A, but the other forms of chlorophyll were just like normal, so they didn't appear to be affected by anything that occurred to them in space. So theoretically, this could be a means of starting to terraform planets, and particularly looking at how we might make ecosystems in extraterrestrial environments, which would be kind of important if we ever want to leave our planet. Next we have some more news going even further from Earth. The Moon. The Moon, and specifically rocks that have been brought back from the Moon. The sample of moon rock has been basically in storage for 50 years, as, the last time we went there was 1972. What the researchers have found is ancient material. The tiny little bit of trilite dust collected in 1972 may actually be a 4.5 billion year old piece of the solar system. Understand, 4.5 billion years old is even older than the moon itself. And researchers were inherently skeptical of their own findings, so they went through and tested everything again, and they found that, actually, this thing is as old as we thought. In fact, that's even more confusing because how the hell do we find it, and where has it come from? The way the researchers have been able to figure out that this is so ancient has to do with using sulfur. Sulfur, in terms of geology, is somewhat more useful than, say, carbon dating, because, in terms of carbon dating, there are a lot of variables and it's only useful in a certain time frame. The sulfur, in terms of geology, is much more useful. And by looking at the isotopes of sulfur, they can figure out roughly the age of something based on ratios. Certain sulfur isotopes compared to other sulfur isotopes basically tells you what's going on in terms of age. However, these samples had no sulfur-33. Now, not having any sulfur-33 is not right. That shouldn't be the case. And this is important, as sulfur-33 is consistent with volcanic degassing, and not finding it there means that this bit of moon soil sample, if we want to call it that, to make it easy, is, not in line with when the moon was being active. It shouldn't be part of the lunar mantle if that was the case. The two major pathways this could have developed is, first of all, the possibility of an interaction between sulfur and ultraviolet light in a very thin atmosphere, which would obviously require the moon to have an atmosphere and very early in the moon's life, but the moon was covered in molten magma at that time, which doesn't really work. You can't have this there and molten magma, it just does not work. This brings the second option, and it's arguably the let's say, leading theory, and that is the moon, when it was being formed, was something smacked off of Earth by a Mars-sized object, and this Mars-sized object could have been what contributed to this particular bit of lunar soil. In terms of Mars, we have news about the habitability of Mars, specifically that Mars may actually have been hospitable to life for much longer than we thought, and this would be useful in terms of figuring out what's gone on. The fact that water once flowed on Mars and there was a reasonably dense atmosphere means that Mars would have been habitable for some time. Now, we say some time for a very important reason, because it's certainly not anymore. Somewhere between 4.2 and 3.7 billion years ago, Mars's water would have basically been disappearing as solar wind slowly stripped its atmosphere away and sucked away all and any water that was available in a reasonably accessible manner. This would have made Mars completely unfit for any kind of habitation. Here is where the new research sort of throws a wrench into those works. The researchers indicate that there would have been fairly substantial periods where Mars would have been suitable for life to grow, and this would have then been, let's say, conducive to life, living and then dying, unfortunately, as, well, the cycle of birth and death on Mars goes through, admittedly, a very long time frame, but goes through a fairly predictable cycle. What makes the researchers say this is that they're looking at the geological strata or layers of soil and rock on Mars, and by looking at dunes within this, particularly the Stimson's Formation, they're able to get an idea about what's going on over a long geological time frame. This is based on Curiosity rover and its ability to, well, be on Mars and take photographs and video of what's happening, and importantly, the environment in which this is happening. The pervasive dry conditions in the Gale crater means these formations were formed during the Noachian period, somewhere between 4.1 and 3.7 billion years ago. And this is a period when there would have been extensive flooding. It's the only way you can really get these formations to develop the way they are. By comparing the formation to similar formations on Earth, they're basically able to definitively say that they formed from interaction with groundwater from a nearby mountain, and that the interaction left behind minerals, gypsum, sulfates, and similar, that you basically find in the Earth's deserts. So they're able to clearly show that, hey, this is what happens on Earth under the right conditions, this is what happens to Mars under the right conditions, they're the same, therefore this is the case. This is important because if we ever get actual humans to Mars, we might be able to find some sort of fossilized remains of life if there was life on Mars in these locations. The formations in question are basically sandstone, which is where we look for a lot of similar remains on Earth because they're really good at retaining that sort of fossilized evidence of life. The last news item we have for you this week is about dark matter, and researchers claiming that they may, well, finally have detected it in our galaxy's glow, which is a really weird way of saying, hey, maybe we found this thing. It's admittedly done in a very complex way, mostly because of how long they've been running this experiment for, let's say. It's an analysis of 15 years worth of data from the Fermi Gamma Ray Space Telescope. What they've found is a unusually high energy gamma ray that cannot be attributed to any known source. It's a glow of sorts. And unfortunately, it's not the kind of glow that you might be thinking of. It's the kind of glow that is, well, radiation. That's, well, gamma ray. Of course it's radiation. The University of Tokyo in Japan has been looking at this and trying to figure out how can we demonstrate that the current hypothetical dark matter exists. Because, well, once you have dark matter and, well, anything else, it collides and it wipes itself out, leading to just energy. Energy is cool, but it's kind of useless if you're trying to prove that something exists. It's kind of like looking at, say, a fuel of sorts, say ethanol, petrol, some other combustible. Burning it, having it produce light, heat, and explosions, and going, hey look, I've proven that it exists, but it's gone now, so I can't show you the thing. This is where we are somewhat skeptical. The collision of dark matter particles is what would cause them to be wiped out, but in being destroyed, they would produce this gamma radiation, and that glow is the gamma radiation they're detecting. So their theory is that we don't have an actual cause that makes any sense other than dark matter. Therefore, we've detected this anomalous output, in a stable manner, and we have no better explanation, so dark matter it is. Their rationale is a little bit better than just that, though. They found it in a reasonably stable diffusion model going towards the center of the Milky Way galaxy, which helps. Second is that the shape, the halo-like structure, matches with the shape that's expected from dark matter interacting in this way, and, well, basically we don't have a better explanation. Now, this is not necessarily definitive proof of anything, but it is one brick in the foundation of proving that, well, in this case, dark matter really does exist. But it's a foundation, and we're not really able to definitively show anything or definitively state anything that's going to be much help quite yet. However, at least in human terms, 15 years of data, which is quite long, is substantial and definitely helps in that respect. In terms of the universe, it's not even the blink of an eye, it's a fraction of a fraction of a fraction of a fraction of a blink of an eye, which means we're not really able to rely on it just yet. But when you combine the sheer volume of power, 20 gigaelectronvolts, and the predicted, range for the interaction of these sorts of particles, and the other variables that they found, there's a good chance that it's the case, but it's also really only matching it to a model that we're theorizing about, which isn't so much help yet. That's all the news we have for you this week. If you found it interesting, consider liking, sharing, and subscribing, and please do post any comments, questions, or suggestions you have below.